All cells die, whether it's due to injury or old age. They have to be replaced, and they have to be replaced exactly as they were before. This requires a very specific set of steps, and we will talk about that in Chapter 10, How Cells Divide, or Mitosis. We're going to begin our study of mitosis with a look at the cell cycle. The cell cycle is the sequence of growth and division in a cell. There are two periods in the cell cycle, interphase and mitosis. Interphase is the longest part of the cell cycle. This is where the cell will grow, will carry out its normal functions, and prepare for mitosis. Interphase is made up of three phases. The G1, called gap phase 1. It's called this because it fills the time gap between the end of the last cell division, or the last mitosis period, and the start of the next DNA synthesis period. Then we have synthesis, which is when DNA is going to be replicated, and then G2, gap phase 2. And this is the time between synthesis of DNA and the actual start of mitosis. We're going to focus our study on eukaryotes because the eukaryotes are the majority of the world's organisms. We're going to start at G1. This is usually the longest period of the three parts of interphase. The G1 phase starts just after a new cell is formed, so the cell begins its growth. The cell also performs its normal functions during this time. At the end of the G1 phase, the cell often rests before moving into DNA synthesis. The resting phase is called the G0 phase. Following the G0 phase, the cell moves on to synthesis. During synthesis, the DNA is replicated in exactly the way we discussed in Chapter 14. In addition, the cell will continue to grow, but the emphasis of energy usage is in DNA replication. Once DNA is replicated, we'll move on to G2. Again, cell growth is continuing, but in this case, the most important energy usage is the replication of the cell's organelles. Also, microtubules will begin to organize to form spindles. These spindles, or spindle fibers, will be used to hold and move the chromosomes during mitosis. Mitosis is the actual division of the cell. When mitosis is complete, if everything goes properly, we should have an exact duplicate of the original cell. The last step in the cell cycle is cytokinesis. Cytokinesis is where the new cell is separated from the original. It's not considered a part of mitosis, nor is it given a separate step. This is another visual of the cell cycle, and it's more representative of the time that's spent in each phase. The time it takes to complete a cell cycle varies between animal cells and organisms. Animal embryonic cells, cells of the embryo, can complete an entire cell cycle in 20 minutes. This is because at that point in their development, they're not actually growing, they're just creating more cells to be used for growth. Fruit fly embryos have the fastest known cell cycle. They can go all the way through the cell cycle in 8 minutes. This is because embryonic cells are focusing on the replication of DNA as they create new cells. Once they mature and they're no longer embryos, the cell cycle takes longer. A typical mammal cell takes about 24 hours. However, a liver cell can take more than a year. The variation in cell cycle length occurs in G1 and G0. Each organism spends more or less time in these phases. Some cells only leave G1 or G0 when an injury occurs and the cell has to be replaced. This is a picture of a working cell, or a cell in interphase. At the top of the cell we have the centrosomes. The yellow things in the middle are called centrioles. The lines around them are spindle fibers, and together they form asters. We'll talk about those when we talk about prophase. We have a nucleus with the nuclear membrane, or the nuclear envelope around it. Inside that we have the nucleolus, and we have the DNA. DNA is in the chromatin, or the long string form. Mitosis is divided into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. They always occur in the same order, and we will look at each one of them separately. Not all biologists will say that prometaphase is its own phase. A lot of them will group what happens in prometaphase with one of the other 2 phases. We're going to look at them separately. Our first phase is prophase. The first thing that's going to happen is the chromatin will thicken and coil into chromosomes. The nuclear membrane will break down, as will the nucleolus. Those materials will come back later. The centrioles will begin to move to opposite sides of the cell, and as they do, those spindle fibers will begin to grow. Biologists aren't really sure what these asters will do, but they think that they might strengthen where the centrioles attach to the membrane. When the centrioles have finished moving, the fibers will stretch between them all across the cell, creating the spindle apparatus. The next phase is prometaphase. This is the transition from prophase to metaphase. It occurs after the nuclear membrane is gone. A second group of microtubules will grow from the poles and attach to the chromosomes at their centromere. This connects each sister chromatid to an opposite pole. This is really important because later on in mitosis, the sister chromatid will be separated using these microtubules. If they aren't correctly attached to the opposite poles, they'll be moved together instead of being separated. This will put both chromatid in one cell and none in the other. The microtubules will begin to move the chromosomes to the center of the cell. This movement is called congression. The movement is not smooth. They don't just slide the chromosomes to the center. They jerk a little bit too far, then they pull them back, and then they move them a little again, until step by step they push them in the line. The goal of prometaphase is to have the chromosomes pretty close to the center of the cell. The next phase is metaphase. The spindle fibers finish moving the chromosomes to the center of the cell. To better understand the placement of the chromosomes, biologists picture a plane perpendicular to the poles, and they call this the metaphase plate, or the metaphase plane. The chromosomes are arranged in a circle along this plate, with the centromeres on the plate and a sister chromatid on each side. If you take a look at the picture at the right, you can see the metaphase plate, and you can see how the chromosomes are set along it. This arrangement ensures that when the chromatid are separated, one will go to each pole, and that means one will eventually go to each of the new cells. Anaphase is the shortest of the mitosis phases. It begins when the centromeres split, creating 2 separate sister chromatid. This happens when the cohesion proteins that are holding the chromosomes together begin to come apart. The sister chromatid are pulled to opposite poles. They're pulled to the pole that their centromere is attached to. This movement happens in 2 ways. In anaphase A, the spindle fibers that are attached to the centromeres begin to be disassembled, making them shorter, and this is what pulls the chromatid toward the poles. In anaphase B, the poles actually move apart. This makes the chromosomes on each side move away from each other, while the cell becomes a little bit more oval, or in the picture, peanut-like in shape. Before we move on to telophase, I want to look at the idea of centromere versus kinetochore. If you remember when I talked about the different forms of DNA and I talked about chromatid and chromosomes, I told you that the centromere holds the sister chromatid together, and then the kinetochore is where the spindle fiber is going to attach. Well, now I've been saying that the spindle fiber is attaching to the centromere. The reason I'm saying this is that kinetochores are actually attached to the centromere, forming a single unit. So when you're describing this, you can say that the spindle fibers are attached to the kinetochores, or you can say that they are attached at the centromere. They are both technically correct. If you want to be exact, you would say that they're attached to the kinetochore because the kinetochore is on the outside and it is attached to the centromere. The last phase of mitosis is telophase. It's the reverse of prophase. The spindle apparatus will be disassembled. The parts will be used to create the cytoskeleton in each of the cells. The nuclear membrane will form around each set of sister chromatid. Because they aren't attached at a centromere anymore, they're back to being called chromosomes. The chromosomes will uncoil and return to the chromatin form. This is important because chromatin can only be replicated when they have been condensed into chromosomes. This will protect the DNA from being replicated when it doesn't need to be. The nucleolus will reappear back inside each nucleus, and then the extra organelles that were made during the G2 phase of interphase will be separated and moved to each cell. Mitosis is over when all of these steps have been completed, but cell division technically isn't done because the two cells are still connected. The last step is cytokinesis, which is the actual separating of the cell into two individual cells. If we look at an animal cell, which is the picture at the top, we're going to look at what looks like a Pac-Man with a Pac-Man mouth. This is an animal cell undergoing cytokinesis. The Pac-Man mouth is the cleavage furrow. The cleavage furrow is created when protein filaments called actins begin to constrict, and you can see those as the creases in the Pac-Man's face. This constricting continues and continues, and the cleavage furrow gets deeper and deeper until finally the two cells separate. In a plant cell, you don't have a cleavage furrow. You have a cell plate. The cell plate forms between the nuclei and builds outward until it fuses to the plasma membrane. When it fuses to the membrane, you have two separate plant cells. This puts all the individual slides together so you can see all the different phases and everything that's happening all at the same time. Mitosis is responsible for the growth, development, and repair of cells. So what we're saying is if an organism needs to grow, if it needs to change, or if it needs to repair itself from injury, the process of mitosis is going to make this happen. Cells control everything that happens, and the control of mitosis begins with a growth factor. A growth factor is a chemical that stimulates the division of cells. If you don't have a growth factor, then mitosis will not happen. Growth factors also stimulate differentiation, meaning it makes the cell take on a specific shape or a specific function. At the end of mitosis, all cells look the same. They're generic. It takes that growth factor to make each cell look different and make each cell begin to function as it's supposed to. DNA is always in control of the cell, including the cell cycle. Scientists believe this control is shown in two ways. The first is that the cell cycle has two irreversible points: the replication of DNA and the separation of the sister chromatid. Once these processes have begun, they have to be completed even if severe mistakes have been made. The other part is the idea of a checkpoint. The checkpoints are used to look at each process for accuracy. If the accuracy is lacking and errors have been found, the cell cycle is stopped. If the errors can be corrected, they are, and if they can't be corrected, just like in the irreversible point control, the cell may choose to opt for apoptosis. The cell cycle's checkpoints also allow the cell to respond to changes in internal and external signals. If, for example, the go-ahead was given to go through mitosis, and now something has happened and the organism is no longer healthy, this checkpoint will allow the cell to stop going through mitosis and do something else with that energy instead. The first of the three checkpoints is the G1S checkpoint. This is the primary point where the cell will decide if it's going to divide or not. It will evaluate external signals like food availability and the presence of growth factors or growth enzymes. If there isn't enough food or there isn't a growth factor, division will not happen. Once the cell has committed to cell division, it must continue, meaning that synthesis must occur and the organelles in G2 must be replicated. Our next checkpoint is the G2 mitosis checkpoint. At this point, DNA replication will be evaluated. If the replication is correct, division will continue and mitosis will start. If the DNA has an error, that error will be corrected. If not, division stops again. Our last checkpoint is the late metaphase or spindle checkpoint. At this point, the cell ensures that all chromosomes are attached to the correct spindle fiber and to the correct pole in preparation for anaphase. Once the cell has started to separate the chromosomes during anaphase, it must continue. When the new cells don't pass a checkpoint inspection, they're dissolved and the molecules are recycled. This is apoptosis. Despite all of DNA's work, sometimes again mistakes happen. When cells lose control of the cell cycle, then they grow uncontrollably and they divide uncontrollably. This creates tumors. Tumors can be benign, meaning they're just an overabundance of cells, or they can be malignant, which is cancer. Cancer cells are cells that have an abnormal size, an abnormal shape, or an abnormal ability. And that ability is to be able to break free from their original place, from the original tumor, and invade other tissues and pass those mistakes on to otherwise healthy tissue, turning them into cancer. And that is how cancer spreads. This is the end of Chapter 10, Mitosis.